did do it.
Sure we can make that work.
Yeah, no worries. Just wanted to let you know.
That's good. Oh, let's get flipped around here. And nothing's gonna happen. Annoying thing that happens.
Oh, that feedback.
Almost there. We've been having connectivity issues all day long. We're almost there. Welcome, everybody.
Mr. Bergeron, can I use the bathroom real quick?
Of course. All right, folks, we're almost there. People coming in anyway. That's good. Oh, I just lost my share though, didn't I? I was in a class during fourth period where the teacher literally could not be heard by the people at home. So hopefully you folks can hear me at home at least.
We can hear you.
Good. Yeah, that was no fun. So everything should be coming up here in just a second. And we'll get started, I hope. Well, you know, maybe it won't. And of course, that's the wrong day. Gets better and better, doesn't it? Well, we might just skip the check-in for the day to save a couple of minutes. All right, well, hopefully now you can at least see the code. go ahead and log in. Everything slows to a crawl this afternoon. There we go. Thank you, thank you. I'm gonna get this process started here. I can always bring the code back up if you can't see it. Just gonna fast forward here a little bit to today. And if you need the code, there it is. So far back. Okay, so far. But we're gonna need some of the lab space too. So do a demo for the folks at home, and the folks here in the building can do something with their own hands. All right, I think we're good now. So welcome everybody. I hope that you can hear me okay and see me okay. Here we are once again. Hybrid, 2 different groups of students in 2 different places, and yet we're gonna try to accomplish the learning together to the best of our ability. So maybe we can do the check-in really quickly here, since it's just a drag. It's Monday, beginning of a new week. Where are you? How are you? Plant your flag, if you will. And just for the sake of simplicity and for the sake of time, notice that you're probably not alone. There's other people who are feeling like you are today. Especially if you're a sea otter or a cat or a dog being hit with a Frisbee. You're certainly not alone. All right. Well, as we look at the agenda, my hope is you had a chance to watch the Edpuzzle video for today on diffusion. I have a really simple lab activity that I wanted to show you. So if you're at home, I'll show you. If you're in the building, I can actually give you a chance to do that on your own. And I do hope that in respect to these Edpuzzle videos, that you're watching them before you come to class. That is the lesson that we want to work with during the limited amount of time that we have together. So I noticed that a lot of people will submit like these videos like after the due date. I get it. Things get busy, things get hectic, but it's going to be harder for you to engage with what we're doing in class if you don't watch those videos ahead of time. Okay, so please, even if the due date, right, precedes your class meeting, which is really important that you're getting those things done before you come to class. So what I'll do to the best of my ability is set up this lab for the folks at home, and the folks here in the classroom will set it up as well. I'll probably try to share my screen with you so that you can see what we are doing in real time. So folks here in the building, why don't you go ahead and find a lab station? If you're already sitting at one, you can stay where you are. And then folks at home, I'll bring over some materials here to the desk so you can see what we are going to be doing here in the classroom. This is going to be tough. We're doing 2 different things in 2 different places at the same time. Sorry, we're doing the same thing in 2 different places at the same time. So what I need you to do is to reach into the beaker, the big beaker in front of you, and grab out into dialysis tubing. Okay, so big beaker on top. centimeter on top. How about this dialysis tubing? All right. So folks at home, I hope you can see this okay. This is a piece of dialysis tubing. Okay, it's flat. Doesn't look much like a tube, but it will in a moment. Okay.
No. It is put it through a machine, so like maybe someone's.
Dialysis tubing, dialysis machine. All right, so what I need you to do is cut off a small piece of string and tie off one of the ends of your tube. So maybe like just tying it off a centimeter from one of the edges. Everything we're going to do today is totally qualitative. It's not quantitative. You cannot do it wrong. I promise. All right, so super exciting, folks at home. Piece of tubing, piece of string. I'm going to tie off one of the ends really simply. Nimble fingers. So, like that. Again, not super exciting.
How much end should we tie?
I'm sorry?
How much end should we tie about here?
You cannot do this wrong. Okay, seriously. You can't screw it up. You can't break it. The worst that could happen is when you pour something inside, it just comes out.
I guess I have it in my mouth.
That would be delicious. All right. Once you got your edge tied off, at this point the tubing might be a little dry. So you might want to re-wet it by dipping it back into the water. But if you rub it between your fingers, the other end, not the end that you tie, it should open up. The tube, right? So right now it's flat. You want to open it up so we can pour stuff inside. You rub it between your fingers till it opens up. It takes some patience, but eventually, wet again. If you want to keep it open, you can the edge of the funnel to put it inside the hole that you create. Carefully. So I've got my little piece of tubing, it's tied off at one end, and I've opened it up. I'm just putting a funnel inside. So eventually I'm going to pour something inside of it. Just like this. You can do this. It starts to dry out, just re-wet it.
No.
Once you got the funnel inside, just hold open, tube inside the side. I'll put it down to the next here. got it? Where's that gonna happen? It's gonna pour something in, it's all just gonna pour out. You cannot do this wrong. Let's take a breath. Let it go. Most importantly, you're being able to open up your eye. All right. Set that aside for the time being. And grab that plastic cup and just fill it with some tap water. I know, super exciting. Cup of tap water. All right, so what you want to do, you got a little transfer pipette and a bottle of iodine. You want to add drops of iodine to your water until it's yellow in color. How yellow? Use what you see on the screen as a guide. That yellow. Again, you can't do this wrong. What if I add too much? It's not going to hurt anything. What if I add too little? It just won't be as clear of a change once we allow for the fusion to occur. You cannot do this wrong. All right. So, little bottle of iodine. I'm sure it's going to be the label or not. We'll transfer a pipette. You're really not missing much, I promise. It looks like pee. It's not pee. Okay. Once your solution is yellow, you can use a pipette to swirl it if you need to. Not possibly do it wrong. What if I call it disappear? Do that? Did that enough? All right, so once you get your water yellow, once you got the iodine, go back to your dialysis bag. We're going to add some starch solution to it. So that's the little flask with the dropper on it. How much? Well, enough to so that you can see it inside the bag, but not so much that you can't tie off the other end. So I have these in my bag. It's always fun. They're all poured in. Yeah. So here. You don't want to use the pipette. Report. Wait on the pump. Oh, that's why everything turned. Yeah, that we just discovered what is going to happen. It starts iodine contact. That's a really big spoiler. Well, you can tie off the other end. Yep. I just tie it tightly. And then come out the other end. Sorry, my first one leaked, so I'm a little bit behind, but just some starch solution inside my dialysis tube. I'm gonna go ahead and tie off the other end with another piece of string. You can tie off the other end with another small piece of string once you're out of your starch. Sure. So, good. Alrighty. So here we are. Both ends are tied. And all I'm going to do, again, you're not missing much, folks. I'm going to take my bag and put it into my beaker that I've added the iodine to. Boop. And then let it sit. Yep, it takes some time. Diffusion takes time. We're going to come back to our seats. Let this sit for about 10 minutes or so. We'll come back to it. Right in the top. Head back to your seats. And we'll come back together. Need to sit for about 10 minutes. All right. So I'm hoping this is going to work, but I'm going to find out really fast if it doesn't or not. You should be able to click on this link that says membrane specificity. It's also on Google Classroom if that is easier for you. People are putting stuff in the chat, but I'm not up at my computer, so I don't know what you're writing. Oh, okay. So when you click on this link, tell me if it says on your end, this assignment has been deleted. If it does, then
It's been deleted.
okay, so we'll just do it together while we're waiting for our diffusion experiment to run its course. Sorry about that, but not a big deal. I don't know why it's been deleted because it hasn't, but suffice to say. All right. So hopefully everybody, so if that link is just telling you that the assignment has been deleted, just look at the Zoom and you should be able to see what you need to see. Okay. Look at the Zoom. It's probably going to tell you, it's called membrane specificity. But the assignment's been deleted, just go back to the Zoom. If you're here in the room, of course, look at the screen. I'll make it bigger so you can read it. This is why it's possible to sit closer to the front. But I can make it bigger. How's that? Can you read that okay? No matter where you're sitting?
Yeah.
Okay. Folks at home, can you see it okay? Okay, good deal. All right, so this online activity is really meant to be review from last week, where we left off last week. So I'm hoping you're noticing a simple representation of the cell membrane. We can see the phospholipids are present. We've got our polar head. I bring out my pen here. Polar head and our non-polar tails. Notice again, the polar heads are facing the inside and the outside of the cell respectively. The non-polar fatty acid tails are making up the interior portion of the membrane. And then we also have this big blue object that is meant to represent a transport protein. So remember, there are different types of proteins that can be found in the membrane. Remember the two different types? They're very general terms to describe the two different types of proteins that can be found within.
Is it peripheral and the other one?
Beautiful, Larry. Larry said peripheral. Sorry, the speaker's not on. Let me switch over. Should have been able to hear you. That's weird. So there are peripheral proteins and there are
Transport? I don't remember.
come on, college level biology. Peripheral proteins and starts with an I, if that helps.
Integral?
There we go. Larry's on fire. Integral proteins. I'm not sure. Actually, I probably know why it's not doing what I need it to do. There we go. Now we'll all be able to hear you, Larry. Peripheral and integral proteins. So is this blue protein a peripheral or an integral protein?
Integral.
Beautiful. Larry's on fire. Who else wants to catch fire? All right. So we've got integral protein. Now, then we have these three different molecules, right? We got a blue, a purple, and a green. Again, if this assignment was doing what it was supposed to be doing, this would be a lot easier. We can see the blue molecules. Well, look at that. They don't go through the membrane, but they do go through the protein. So again, not through the membrane, but through the protein. The green molecules do not go through the membrane, and there's no way for them to go through the protein either. Purple molecules, similar to the green, they do not go through the membrane, nor is there a path for them through the protein. Okay, so this is already showing us in terms of membrane specificity that this particular integral membrane protein is specific to the blue molecule. It allows for the passage of the blue molecule, but not the purple or the green. But none of the three molecules as presented can cross the membrane by themselves. Okay. So as we get started here, why might that be the case? Blue, green, or purple, it doesn't matter which one you're thinking about. Why might that molecule not be able to cross the membrane all by itself? What might be true about it?
Repelled from the membrane.
Okay, it's repelled. Okay, let's build on that idea. If it's being repelled by the membrane, why might it not be able to cross the membrane?
Enough energy?
Okay, so it might not have enough energy, certainly a possibility. So that would be probably a direct consequence of the temperature of the environment in which this model of the membrane is found. But then I think I heard Isaac, you want to say it again real quick?
It's large.
It's too big, right? If the molecule is too big to cross the membrane, it's not going to be able to fit in between those gaps, going back to last week again, that spontaneously form between the phospholipids and the proteins and the rest of the mosaic membrane, right? That form as a result of all of these molecules moving and bouncing off of one another. In fact, if you'd like, as a refresher, if I go back to the notes here real quick, I hope it'll be quick. You might remember this. Yeah. Man, everything is slow today. Remember, all of these molecules are in constant motion, right? Whew, it's laggy. I'm sorry about that. And because the molecules are in constant motion, these gaps form between the molecules, and sometimes those gaps are large enough to let small molecules cross the membrane. But going back to what Isaac just said a moment ago, no way for large molecules to cross through those same gaps. Okay, so maybe the blue, the purple, or the green represent really large molecules. What else might be true about the molecules?
They may be not polar, non-polar.
Well, if they're non-polar, what would we expect them to be able to do? Would non-polar molecules be able to cross the non-polar interior of the membrane? Does like dissolve like? Going back to chemistry. We have to go fast, folks. I need you to be on point.
Yes, they would, but they wouldn't be able to get to the polar part of the membrane.
Unless those gaps, right, that form spontaneously allow for a passageway for those non-polar molecules to move across. Because once they got through that little gap at the polar side of the phospholipid, they can quickly dissolve into the non-polar interior And as long as a gap opens on the other end, the molecule can cross to the other side of the membrane. Okay, but if the molecule is polar, we're going to have a problem because that polar molecule is not going to be able to easily cross the non-polar part of the membrane. So if the molecule is too big, or if it's polar, it's likely not going to be able to cross the membrane very quickly. The other factor that's worth considering in terms of whether or not a molecule can cross the membrane is whether or not it's charged. So if it's an ion, positively or negatively charged, think of all the different ions you learned about in chemistry. Of course, you memorize them all, right? And their charges, their names, all that good stuff. Of course, you haven't memorized them all. You know whether or not a particle is an ion, +1, +2, +3, -1, -2, -3. Whatever the charge in the ion is, it's probably not going to be able to cross the membrane by itself. But it's okay because in many instances, not all, but in many instances, there's going to be a transport protein available to move that particular particle across the membrane. Okay? So this first part of the activity is just to kind of think about some of the different reasons why a molecule might not be able to cross the membrane all by itself. Does that bring up any questions for you, whether you're at home or here in the classroom? I'm going fast. Okay. No one's screaming at me. All right. So in a perfect world, we'd be able to do this on our own. But we can quickly do it together. If you're on Zoom at home, you can answer it in the chat. Okay. If you're here in the classroom, you can read the options and tell me what to click. Or you can just yell out what you think the correct answer is once you think you have it. Well, what do transport proteins do in general?
Transports protein, transports molecules.
They transport molecules. So which of these choices do you think is going to be the most likely to be correct? A? What do you think? Worst that can happen is we get it wrong, right?
Yeah. This is the AP test.
This is not the AP test, this is practice, right? So indeed, those proteins are going to allow for molecules to move in and out of the cell, in this case, just the blue molecule. Which probably means it's either too large or too polar or charged to cross the membrane by itself. Okay. All right, so what could that transport protein be doing so as to allow a molecule to cross that wouldn't be able to cross on its own? How might it be deciding? Which choices do you think make the most sense here?
Size or shape, because it can see color because you can't see the photon lights reflecting off the molecule.
Can't see color. That doesn't make any sense. I agree.
I think charge color is too.
Charge too,
Yeah,
but not color.
no, not color.
I agree.
Oh, wow.
Agree. Right. So that protein is determining what type of molecule crosses based on how big it is, its charge, right? And shape could certainly be important as well. All right. Again, review here. Thinking about that transport protein, the blue molecule that you see on the screen, if you think about the tips, the left and right hand tips, are they hydrophilic or hydrophobic?
Hydrophilic?
Hydrophilic, right? Those are the polar sides of the phospholipid. That's the polar side of the membrane. We expect the protein to be interacting with the polar side of that protein as well. Moving along here. What do you think? Think of that animation, right? Are the forces that hold together the membrane super powerful?
No.
If they were, we expect the membrane to be more solid and not fluid.
So it's probably A. So the first part would be
remember there was a question we considered last semester when polypeptides folded into proteins, right? And we expected our little model of our protein to fold within an aqueous, polar, watery environment. And as a consequence, all the polar charged amino acid side chains were facing outward, while the non-polar side chains were facing inward. But if we switched, there's even a multiple choice question that we considered. If we flipped the environment and made the environment non-polar instead of polar, everything in that protein got flipped inside out. So the same thing would happen to the membrane, right? If that membrane was not forming in water and instead formed in a non-polar environment, we expect the tails to be facing outward and the polar heads to be facing inward.
When we cover ourselves in oil, we don't turn inside out.
What you do on the weekends is totally up to you. Okay.
I do cooking, okay?
You cover yourself in oil?
I get olive oil in my hand sometimes. Cooking is messy.
Well, then it gets very specific very quickly where the layers on the outermost, the cells in the outermost layers of your skin aren't even alive anymore. It's just dead cells connected by protein. So the membrane is actually, for the most part, dissolved at that point. It's already gone. So there's nothing to flip inside out would be the short answer to your question or point. All right, just one more to consider here. You're absolutely right about that one. This one doesn't have a correct answer to choose, but brings us back to some of the learning we did earlier last week. If a protein wasn't present, how might a molecule cross the membrane? Why don't you quickly, before you share, Isaac, I think all of us are close enough to somebody where we can share briefly. Okay. And folks at home, what I can do, hopefully there's somebody here. There's a lot of you here learning from home today. If you're willing to share with me what you think might be responsible for the motion of that molecule or the movement of that molecule across the membrane, I'm the only person that can hear you. Or you can put it in the chat. There's no protein present. How might the molecule cross the membrane? It's definitely something to do with diffusion. Give you another hint. If that protein wasn't present, if that big blue cylinder wasn't present, how might molecules cross the membrane?
Part of themselves.
Sorry?
Like, what if you shoot part of yourself through at a time?
Well, they can't, they're not sentient beings, right? They don't know how to speed up or when to speed up.
Could they squeeze through?
Again, it comes back to this notion. I know we're going fast, and this can be tough. I'm hoping you're going to ask questions if you don't understand. The molecules that make up a cell membrane, the phospholipids, the protein, the cholesterol molecules, all those different components, they're in constant motion. Okay, the membrane is fluid. But because the molecules that make up the membrane are in constant motion, there are these gaps that spontaneously form within the membrane. And those gaps are what are responsible for allowing particles, as long as they're small, and preferably nonpolar and uncharged, to cross the membrane. Okay? You don't have to have a protein present in order to move those molecules across. Certainly having proteins present is a far more efficient mechanism for membrane transport. But it would have been possible for the earliest forms of life to still move molecules across their membrane because the molecules that make up the membrane are in constant motion. And the gaps that form as a result would have provided a passageway for those molecules to get across.
So they also do like when an amoeba eats another amoeba and then like they go around and then like they like squish their membrane and then like press it in, so then they just have to like absorb the thing inside.
Yeah, it's a great question. If you didn't hear what Isaac said, he's thinking of like amoeboid It's a process called endocytosis. What's required, though, is a cytoskeleton that pushes the membrane outward that allows for the membrane extension necessary to surround whatever it is that the amoeba is eating. The earliest cells on Earth wouldn't have had that cytoskeleton, so they wouldn't have been able to do that particular mechanism. They would have been really just relying on molecules colliding with the membrane, and if a gap formed at just the right time, a passageway forming that would have allowed the molecule to cross. But later on in life, when that part of the cell became a thing, then that would have been possible, for sure. But not in the earliest forms of life. Yes, Larry, absolutely. People can't see what you wrote in the chat, but yes, it goes back to the idea that the membrane is fluid, there are spaces between the molecules, and as a consequence, molecules can cross the membrane. All right. All right, I think we've got about 5 minutes left. So folks at home, I'm gonna surprise you with the results of the experiment that we started at the beginning of class in a moment. Folks here in the classroom though, go ahead and grab your cup really quickly. You can bring it back to your seat. And the change should be pretty obvious. Ready for the big reveal? Blue. Is it okay? All right. So what I'm hoping you're noticing is that the starch solution that you poured in at the beginning is now blue. But wait a minute. We're thinking about the video that we watched before coming to class today about diffusion. What happened?
Squeeze rid of osmosis.
Well, osmosis is a different biological concept. We're actually not investigating that yet.
Osmosis.
It's diffusion. Okay, so what diffused in? The iodine molecules diffused in. Okay, so what must that mean about their molecular size?
They're relatively small compared to the size of the plastic molecules.
Absolutely. So technically, Isaac, if you can't hear what he's saying, the dialysis membrane, the dialysis tubing, is obviously meant to represent the cell membrane, but is not a true cell membrane. Okay. But regardless, the iodine molecules, and if you get really specific, iodine, I2, it's just composed of 2 atoms. It's about as small of a molecule as you can possibly get, right? So the iodine molecules moved into the bag because there's a higher concentration of iodine on the outside of the bag compared to the inside of the bag. Right? That's diffusion. What about the starch? Did the starch move out of the bag? You might be wondering, how do you know?
You can't really tell because the solution didn't change slightly lower from it's still liquid.
But on the outside, so in that yellow solution, did it turn blue?
Not at all.
Not at all. So what does that tell you about the starch molecules? Because there's a lot more of them inside the bag than outside the bag. Say it louder. The starch molecules, right? And if you remember from last semester, starch is a polymer of glucose molecules, usually thousands of glucose molecules long, right? It's so big, there's no way that it crossed the membrane on its own, right? So the starch stays inside, even though there's a higher concentration of starch on the inside of the bag, it was too big to cross the membrane on its own, right? So this is obviously really simple and really quick, but I'm hoping you're seeing that diffusion is real, right? We can observe the effect of diffusion in a simple experiment like this one, all right? All right, everything can be dumped out. If you want to save the dialysis tubing, you can. Otherwise, it can be thrown away. Pretty amazing when you think about it.
funny because the water molecules are also smaller than the plastic molecules probably.
So technically, if you weigh the bag before you put it into the solution, and you weigh it after you took it out of the solution, there should be an increase in mass, because water moves in. But that's for later.
So if we left it longer, would the color get deeper and deeper?
The saturation point, for sure. I would say it's just about as blue purple. All right, folks, that was the bell. So thank you for sticking with us. I'll see you in a couple of
The bag was
days.
full with no iodine in it.
If the bag was,
Thank you.
only if there was enough pressure from the inside moving outward to prevent the movement of molecules inward.
Thank you.
Thank you, Mr. Bergeron. Have a good day. Thank you.
See you, everybody.
Zach, that was all hacks.
I need to figure out. You're welcome.